Welcome back to the deep dive. Today, we are strapping into the centrifuge and spinning right into what feels like the future of volcanology, a future powered pretty much entirely by generative deep learning.
It's an incredible topic. It really is a story about how AI is starting to overcome some of the fundamental constraints that nature imposes on us.
It really is. So our sources today detail this huge computational breakthrough. Scientists aren't just copying biology anymore. They're designing these tiny, custom-made proteins engineered to do multiple things at once, specifically for next-gen vaccines.
And in this case, for respiratory syncytial virus, or RSV.
Exactly. And for you, our mission today is to really unpack the mechanics of this. How did researchers get past the old methods to build these small proteins that can hold, three complex, really irregular viral features all at the same time?
Yeah, and before we get into the nuts and bolts, we should probably define a few key terms. They're pretty foundational to really grasping the scale of this thing.
Okay, let's do it.
So first, we're talking about de novo protein design. This isn't about finding a protein that already exists and, tweaking it a bit.
Right, this is building from scratch.
Completely from scratch, generating totally novel structures that just don't exist in nature's vast library. It's a whole new ballgame.
It's like the ultimate biological Lego set, but you're inventing the bricks themselves as you go.
That's a great way to put it, and that matters because the target itself is so specific. That brings us to the epitope.
Okay.
That's the particular, often really irregular molecular shape on the surface of a virus, like RSV, that your antibodies have to recognize to do their job. It's the precise thing we need to teach the immune system to go after.
So we're building a brand new vehicle, that's the protein scaffold, and it's designed to perfectly display this target, the epitope.
Precisely. And that whole package, the vehicle plus the target, when you use it in a vaccine, that's your immunogen. It's the thing that triggers the immune response.
And the core claim in our sources is that researchers successfully designed these tiny immunogens, right? We're talking under 130 amino acids, so incredibly compact.
So small. And yet they present three distinct epitopes from the RSV fusion protein, RSVF.
Which is the protein RSV uses to actually fuse with our cells, so it's the perfect target. This whole thing just demonstrates the power of deep learning to solve these incredibly complex design problems that they were considered impossible before.
It's a fundamental shift. You go from searching for a decent enough match in a database to just creating the perfect, custom-tailored solution from thin air.
Okay, so let's unpack that. Let's start with the first challenge they had to solve. Just getting one single epitope to work.
Right.
Because historically, if you wanted to graft a piece of a virus onto a protein, you were kind of stuck with what was available in the protein data bank, the PDB.
The PDB is, it's basically the known universe of protein structures. It's an amazing resource, but it has this huge limitation. If the viral part you want to display, that epitope, doesn't really look like anything that's already in the PDB.
You're out of luck. It's like you need a very specific curved triangular Lego piece, but you've only got standard rectangular blocks.
Exactly. And that meant most engineered proteins could only handle one simple job, one motif. But in nature, proteins almost always have multiple functional sites, so we were really hamstrung by that.
Which brings us to their first test case, a really notorious challenge in this field, RSVF Site V.
Ah, yes, Site V.
This specific motif had, and I'm quoting here, eluded traditional design methods. For exactly that reason. It just wasn't similar enough to anything in the known library of proteins. It was that impossible triangular piece.
And that's where they brought in the heavy computational artillery. They needed a way to, break free from the PDB's limits. And they did it by combining two key deep learning tools.
Okay, let's translate those.
Yep.
What were the tools and what do they actually do?
So first, they used Rosetta Fold, or RF, for something they call structural hallucination.
Hallucination.
It sounds a bit wild, right? But just think of it as an AI that can dream up completely new, stable 3D shapes. It's the engine that's generating the blueprints for these novel scaffolds.
So it dreams up the shape. But what about the actual, material to build it?
That's where the second tool comes in, protein MPNN. Once RF dreamed up a promising new shape, that blueprint gets fed into protein MPNN. Its job is to design the perfect amino acid sequence, the actual chemical code, to make sure that dreamed-up shape is stable and will actually fold correctly in the real world.
So it's this incredible feedback loop. Dream up a shape, then write the perfect chemical recipe to make that dream a reality. So how did the results look compared to the old methods that kept failing?
The results were immediate and just, a drastic improvement. When they actually made these single epitope designs, the structural accuracy was phenomenal. One of them, RSVFV1, was just exceptionally precise.
I saw the metric for that in the sources. A root mean square deviation, an RMSD, of only 1.05 angstroms. Now, for the rest of us, what does 1.05 I really mean?
It means it's basically perfect. That's atomic level accuracy. The RMSD measures the average distance between where the atoms were predicted to be and where they actually ended up. A number that low means the AI's blueprint was virtually identical to the real-world protein. It's just a stunning validation.
Okay, so the shape was a perfect mimic. But that only matters if it's functional, right? Did it actually bind to the antibody it was supposed to?
It absolutely did, and this is where the functional improvement was just staggering. They measured binding with a value called KD, and lower is better. It means a tighter bond.
And what did the numbers say?
Well, these new designs had KD values between 54 and 241 nanomolar. And here's the kicker. Compared to all the previous attempts on this exact same difficult epitope, this was a 20-fold improvement in binding strength.
Wow, 20-fold.
Yeah, it just proved that deep learning could crack these single epitope problems that traditional methods just couldn't touch.
Okay, so conquering one tough epitope is a huge deal. But for a really good vaccine, you want to train the immune system against multiple weak spots on a virus. So the next step was to, just dramatically escalate the challenge.
That's right. They aimed to go from one epitope to 3 distinct non-overlapping epitopes from RSVF sites 2, 4, and VL displayed on one single tiny scaffold. This is where the engineering complexity just skyrockets.
And why go through all that pain? Why triple the
It's
complexity?
all about efficiency. The goal was to create an immunogen with a higher ratio of the good stuff, the viral surface compared to the scaffold holding it together. It's about maximizing the training signal for the immune system and minimizing the noise.
So to do this, to put off this triple scaffold, they couldn't just use the standard RF hallucination tool. They needed something more advanced.
Right. They used a specialized technique called RF joint-2 inpainting. It's sort of an evolution of the Rosetta fold network, but it's adapted for the specific problem of connecting multiple separate pieces.
Let's focus on the key flexibility here, because this seems to be where the AI really breaks away from the old constraints.
This is what's so fascinating. They could input the templates for all 3 epitopes, Site 2, Site 4, and Site V, without telling the AI how they should be arranged relative to each other. In the past, you would have had to dictate the exact spacing and orientation found in the native virus. But that native arrangement is often huge and way too complex to fit onto a tiny, efficient scaffold.
So the AI wasn't forced to copy nature's giant, complex blueprint. It was allowed to find the most efficient arrangement on its own.
Exactly. And that flexibility was the key.
Yeah.
It let the model generate this huge diversity of possible scaffolds and find a unique solution that could physically hold all 3 pieces, no matter how they're arranged on the actual virus. It opened up this completely new design space.
Okay, let's walk through that computational pipeline, because it's a masterclass. Step 1 was backbone generation.
Right. RF joint-2 inpainting handled that. It simultaneously built the connecting bits, the scaffold, and it chose the best relative positions for the 3 motifs in, what had to be a non-native orientation. It just figured out the optimal geometry.
And once it had that geometry, that backbone, what was step 2?
That was sequence design, which was handed back to protein MPNN. Its job was to design the optimal amino acid sequence for that brand new scaffold to make sure it was stable and would actually fold into that very specific complex shape.
And finally, a really rigorous filtering step. I have to imagine when you're connecting 3 irregular pieces like this, you generate a lot of, jump proteins that don't work.
Oh, absolutely. The filtering was meticulous. They had to check for functional accessibility. So they did this essential computational check. They basically tried to dock the 3 target antibodies onto the design to see if they'd fit.
See if there were any physical blockages.
Exactly, what we call steric clashes. If the antibody couldn't physically reach the epitope because the scaffold was in the way, the design was immediately thrown out. That filtering was vital.
Okay, so that's the theory, the computational design. The big question is always, did these custom triple function proteins actually work in the real world?
They did. The in vitro validation was fantastic. The designs RSVF multi 1 through 4 were incredibly robust. They showed really high thermal stability, minimal unfolding, even when you heated them up to 90 degrees Celsius.
So they're tough little proteins.
Yeah.
But did they do their triple job?
Passed with flying colors. All 4 of the successful candidates bound to each of the 3 different site-specific antibodies. So motivizumab for site 2, 101F for site 3, and RSV90 for site V. It just confirmed they were successfully presenting all three epitopes at once.
I have to imagine that when you go from designing one perfect thing to scaffolding three, there's a huge potential for the accuracy to drop. Did the structural fidelity hold up?
That's a great question, and the proof is just astonishing. They got X-ray crystal structures for two of the best ones, and they confirmed extremely close agreement with the computational models. Again, a backbone RMSD of around 1.4 of A.
Wow. Even with all that complexity.
Even with all that complexity.
So the AI designed something incredibly complicated. It was made, it folded exactly as predicted, and it worked. But here's where it gets really mind-bending for me. The structural novelty. The sources say the overall folds of these designs are quite unusual.
And this right here is probably the biggest takeaway for the whole field. When they compared these new designs to the entire protein databank, that whole known universe of proteins, the closest natural structures had what's called a TM score of 0.46 or lower.
Okay, stop there. Unpack the TM score. What does that number mean?
So the TM score measures the similarity of two protein folds. A score of 1.0 means they're identical. But the crucial part is that a score of 0.5 is generally the line in the sand for a completely different protein fold.
So a score below 0.5 means when you compare these to everything we know, the closest match was basically a stranger.
Precisely. It means the AI wasn't just remixing things it had seen before. It generated truly novel fold structures that have never been seen in nature or characterized by science, just to solve this very specific, very constrained problem.
So the computer created something entirely new, out of necessity, to do the job. That's not imitation. That's a form of creativity.
It is. And that creative solution brings us back to the core biological advantage. Why do this instead of, say, a cocktail vaccine? It comes down to what you could call the decoy surface.
The decoy surface. Tell us about that.
Think of it like this. With a traditional single-epitope design, maybe only 20 or 30% of the protein you're injecting is the relevant part, the epitope. The other 70% is just the scaffold holding it up.
So it's junk mail for the immune system.
It's total junk mail. Your immune system wastes a ton of energy making antibodies against that scaffold, antibodies that won't do anything to fight the actual virus. That's the decoy problem.
And these new designs.
These new multiepitope designs are so efficient that more than 50% of their surface is the desirable stuff, the viral parts. They massively cut down on the junk mail.
So did that theoretical advantage actually translate to better results in vivo, in mice?
It did, and the comparison was really stark. The single epitope design, the one that was structurally perfect, it confirmed the decoy problem. It elicited a strong immune response to itself, but it produced no detectable neutralizing activity against the actual virus.
So most of the antibodies were hitting the useless scaffold. The structural perfection didn't matter.
Exactly. But the multi-epitope scaffolds were a different story. They showed moderate levels of cross-reactive titers against the full RSV protein. And critically, these titers were improved by several orders of magnitude over that failed single-epitope design.
A massive jump in actual utility.
And the ultimate goal was achieved. One of the three epitope immunogens, RSVF multi-1, successfully demonstrated physiologically relevant neutralization titers against the virus. They created a single, custom-built component that could actually train the immune system to fight.
So what does this all mean for the future of making vaccines, for responding to new threats?
Well, it offers a really powerful alternative to complex cocktail vaccines. Instead of making and validating three separate proteins and mixing them, you make one that does all three jobs. It could slash manufacturing costs, simplify quality control, and just dramatically speed up the pipeline. It's a huge step towards a kind of plug-and-play vaccinology.
So to summarize the takeaways here, we've seen this incredible power shift. Deep learning lets scientists custom-build stable, multifunctional proteins, and they're no longer limited by what biology has already made. They're creating brand new solutions.
And the implications go so far beyond vaccines. This ability to put multiple functional sites onto a single, stable de novo scaffold. Think about what that means for designing advanced enzymes or sophisticated biosensors or next-generation therapeutics that need to bind to multiple targets.
So the real success isn't just in copying nature accurately, which AI is already good at. It's in the AI's ability to create fundamentally new structures, new folds that have never existed, just to solve a really hard problem.
Exactly. This really signals a new era where engineered biology is limited less by what we can find in nature and more by the creativity of the problems we pose to these models. The future of designer biology is here, and we're no longer limited by what exists, but by what we can computationally imagine.